All right, let's dive into the plant kingdom. So in the previous chapter, we looked at Whitaker's 5-kingdom classification from 1969. Now in this chapter, we're focusing specifically on kingdom Plantae, which includes 5 major groups: algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. These are the groups we'll be exploring in detail. Now here's something important. Our understanding of the plant kingdom has actually changed over time. Earlier, things like fungi and organisms from Monera and Protista with cell walls were included in Plantae, but now they've been moved out. A classic example? Cyanobacteria, which used to be called blue-green algae, aren't actually considered algae anymore. So the plant kingdom is more refined now than it used to be. So when botanists started classifying plants, they used what we call artificial systems first. These were based on just a few obvious characteristics like leaf shape or flower color. And the problem was they often separated species that were actually closely related. Then came natural classification systems, which were much better because they looked at internal features to things like anatomy, embryology, and even the chemistry of the plant. George Bentham and Joseph Dalton Hooker developed an important natural classification system for flowering plants. Today, we use phylogenetic classification systems, which are based on evolutionary relationships between organisms. The assumption is that organisms in the same taxa share a common ancestor. And we have some really cool modern tools to help us classify plants. Numerical taxonomy uses computers to analyze hundreds of characteristics. Cytotaxonomy looks at chromosome numbers and structure. And chemotaxonomy uses chemical constituents to resolve confusions. Technology has really revolutionized how we classify plants. Let's start with algae. Algae are pretty fascinating. They're chlorophyll-bearing organisms, which means they can photosynthesize. They have a simple thalloid body structure. Basically, they're not differentiated into roots, stems, and leaves. They're autotrophic, so they make their own food. Most algae are aquatic, living in both freshwater and marine environments, but you'll also find them in other habitats like on moist stones, in soil, and even on wood. Some even live in association with fungi. Those are lichens, or with animals like on sloth bears. Algae come in all shapes and sizes. You've got colonial forms like volvox, filamentous forms like Ulithrix and Spirogyra, and then these massive marine forms called kelps. Their sexual reproduction is interesting. It can be isogamous, where the two gametes are similar in size, and isogamous, where they're different sizes, or oogamous, where you have one large non-motile female gamete and a smaller motile male gamete. Here's why algae are so important. They fix at least half of the total carbon dioxide on Earth through photosynthesis. That's huge. They also increase dissolved oxygen levels in their environment. About 70 species of marine algae are used as food, species like Porphyra, Laminaria, and Sargassum. And we get commercial products from them too, like agar from Gelidium and Gracilaria, which we use to grow microbes and make ice creams and jellies. There's also algin from brown algae and carrageen from red algae. Even chlorella, a unicellular alga rich in proteins, has been used as a food supplement by space travelers. Algae are divided into 3 main classes based on their pigments and stored food. First up, we have Chlorophyceae, the green algae. Second, there's Phaeophyceae, or brown algae. And third, we have Rhodophyceae, the red algae. Let's look at each of these in detail. So green algae, they're called that because they're usually grass green, and that's due to the dominance of chlorophyll A and B pigments. These pigments are located in definite chloroplasts. Most green algae have these storage bodies called pyrenoids inside their chloroplasts. These pyrenoids contain protein and starch. Their cell wall structure is interesting too. There's an inner layer of cellulose and an outer layer of pectose, making it pretty rigid. The chloroplasts in green algae can have all sorts of shapes: discoid, plate-like cup-shaped, spiral, or ribbon-shaped, depending on the species. Some common examples you should know are Chlamydomonas, volvox, Ulithrix, Spirogyra, and Chara. These are the ones you'll see most often in studies of green algae. Brown algae are found primarily in marine habitats. Their size variation is incredible. They range from simple branched filamentous forms like ectocarpus to these massive profusely branched forms called kelps, which can reach heights of 100 meters. That's as tall as a 30-story building. They have chlorophyll A and C, plus carotenoids and xanthophylls. The brown color comes from the xanthophyll pigment fucoxanthin, and the amount of this pigment determines whether they're olive green or various shades of brown. Brown algae store their food as complex carbohydrates, specifically laminarin or mannitol. Their body structure is pretty organized. They have a holdfast that attaches them to the substrate, a stalk called the stipe, and a leaf-like photosynthetic organ called the frond. The cell wall has cellulose, but it's usually covered with a gelatinous coating of algin on the outside. Common examples of brown algae include Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. These are the major representatives you'll encounter. Red algae get their name from the predominance of the red pigment phycoerythrin in their bodies. The majority of red algae are marine, and you find greater concentrations of them in warmer areas of the ocean. Red algae are pretty adaptable when it comes to depth. They occur both in well-lighted regions close to the water surface and also at great depths in oceans where relatively little light penetrates. They store food as Floridian starch, which has a structure very similar to amylopectin and glycogen. Red algae reproduce sexually through oogamy, and their post-fertilization developments are quite complex. They reproduce asexually by non-motile spores. Note that both their spores and gametes are non-motile, which is different from other algae. Common members include Polysiphonia, Porphyra, Gracilaria, and Gelidium. Remember, we get agar from Gelidium and Gracilaria. Now let's move on to Bryophytes. Bryophytes have this cool nickname. They're called the amphibians of the plant kingdom. Why? Because just like amphibians in the animal world, these plants can live in soil. They're terrestrial, but they're still dependent on water for sexual reproduction. You usually find them growing in damp, humid, and shaded areas, commonly in the hills. The plant body of Bryophytes is more differentiated than algae, but less so than higher plants. The main plant body is haploid and produces gametes, so we call it gametophyte. This is the dominant phase you see. Now, after fertilization, they produce a sporophyte, but here's the key thing. The sporophyte is not free-living. It stays attached to the photosynthetic gametophyte and actually gets its nutrition from it. It's like a dependent generation. Bryophytes don't have true roots, stems, or leaves. Instead, they have structures that look like them, root-like, leaf-like, and stem-like structures, and they're attached to the substrate by unicellular or multicellular rhizoids. The bryophytes are divided into two main groups, liverworts and mosses. They play an important ecological role in plant succession on bare rocks and soil. Let's talk about how bryophytes reproduce. Their sex organs are multicellular, which is an advancement over algae. The male sex organ is called an antheridium, and it produces these biflagellate antherizoids. That means they have two flagella for movement. The female sex organ is called an archegonium, which is flask-shaped and produces a single egg. The antherizoids are released into water, where they swim to reach the archegonium and fuse with the egg. After fertilization, the zygote doesn't undergo reduction, division immediately. Instead, it develops into a multicellular sporophyte. The sporophyte is differentiated into three parts: a foot, which anchors it to the gametophyte, a seta, which is like a stalk, and a capsule at the top. Inside the capsule, some cells undergo meiosis, reduction division, to produce haploid spores. These spores are then released and germinate to produce a new gametophyte, completing the life cycle. So you have this alternation of generations between the dominant gametophyte and the dependent sporophyte. Liverworts typically grow in moist, shady habitats. You'll find them on banks of streams, on marshy ground, in damp soil on the bark of trees, and deep in woods. The plant body of a liverwort is thalloid, meaning it's a simple flat structure, and it's dorsiventral, meaning it has distinct upper and lower surfaces that are different from each other. Marcantia is a classic example. The thallus is closely pressed against the substrate. Liverworts have a neat method of asexual reproduction using structures called gemmae. Gemmae are green, multicellular asexual buds that develop in small receptacles called gemmacups located right on the thallus. These gemmae can detach from the parent body and germinate to form new individuals. It's a very efficient form of vegetative propagation. The life cycle of a moss is particularly interesting. The predominant stage is the gametophyte, which actually has two distinct stages. First, there's the protonemous stage, which develops directly from a spore. It's creeping, green, branched, and frequently filamentous. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. This stage consists of upright, slender axes bearing spirally arranged leaves, and they're attached to soil through multicellular, branched rhizoids. The sporophyte in mosses is more elaborate than in liverworts. It still has the foot, seta, and capsule, but they're more developed. The capsule contains spores and has an elaborate mechanism for spore dispersal. Common examples of mosses include Funaria, Polytrichum, and Sphagnum. Speaking of sphagnum, this moss is economically important. Species of sphagnum provide peat, which has been used for a long time as fuel and as packing material for transporting living materials. This is because of their amazing capacity to hold water. Mosses, along with lichens, are also the first organisms to colonize bare rocks, which makes them ecologically very important for soil formation. Now we're moving up in complexity to pteridophytes. The pteridophytes include horsetails and ferns. Here's what makes them evolutionarily significant. They're the first terrestrial plants to possess vascular tissues, specifically xylem and phloem. This was a major advancement that allowed plants to grow taller and transport water and nutrients more efficiently. Unlike bryophytes, where the gametophyte is dominant, in pteridophytes the main plant body is the sporophyte. And this sporophyte is differentiated into true roots, stems, and leaves, all with well-differentiated vascular tissues. The leaves can be small, which we call microphylls like in Selaginella, or large called macrophylls like in ferns. The sporophytes bear sporangia. These are the spore-producing structures, and they're subtended by leaf-like appendages called sporophylls. In some cases, these sporophylls are organized into distinct compact structures called strobili or cones. You see this in Selaginella and Equisetum. These are sites of spore production. The sporangia produce spores by meiosis in spore mother cells. When these spores germinate, they give rise to small, inconspicuous, multicellular, free-living, mostly photosynthetic gametophytes called prothallus. Now here's an important limitation. These gametophytes require cool, damp, shady places to grow. This requirement restricts where pteridophytes can successfully reproduce. The prothallus bears male and female sex organs, antheridia and archegonia respectively. And just like bryophytes, water is absolutely required for reproduction because the male gametes, called antherizoids, need water to swim from the antheridia to the mouth of the archegonium. Once fusion occurs, the zygote develops into a new sporophyte, which is the dominant phase you see as the fern plant. Most pteridophytes are homosporous, meaning all their spores are similar in kind. But some genera like Selaginella and Salvinia are heterosporous. They produce two kinds of spores, large megaspores and small microspores. The megaspores germinate to produce female gametophytes, and microspores produce male gametophytes. What's really interesting is that in these heterosporous pteridophytes, the female gametophytes are retained on the parent sporophyte for variable periods, and the development of zygotes into young embryos takes place within these female gametophytes. This is actually a precursor to the seed habit, which is considered a really important step in plant evolution. Pteridophytes are classified into 4 classes. First, Psilopsida, which includes Psilotum. These are the most primitive. Second, Lycopsida, which includes Selaginella and Lycopodium. These are the club mosses. Third, Sphenopsida, represented by Equisetum. These are the horsetails. And fourth, Pteropsida, which includes the true ferns like Dryopteris, Pteris, and Adiantum. These are the most diverse and familiar Pteridophytes. Let's move on to Gymnosperms. The name literally tells you what they're about. Gymnos means naked and sperma means seeds. So gymnosperms are plants where the ovules are not enclosed by any ovary wall. They remain exposed both before and after fertilization. This is their defining characteristic. The seeds that develop after fertilization are not covered. They're literally naked seeds. Gymnosperms include medium-sized trees to very tall trees and some shrubs. One of the most impressive examples is the giant redwood tree sequoia, which is one of the tallest tree species on Earth. Gymnosperms have some interesting adaptations. Their roots generally form taproot systems, but in some genera, they have special associations. In Pinus, for example, the roots have a fungal association called mycorrhiza. In Cycas, there are specialized coralloid roots that are associated with nitrogen-fixing cyanobacteria. That's how they get nitrogen. The leaves are well adapted to withstand extreme conditions. In conifers, the needle-like leaves reduce surface area, and they have thick cuticles and sunken stomata to help reduce water loss. These are xerophytic adaptations. Gymnosperms are heterosporous. They produce both microspores and megaspores. The sporophylls that bear these spores are arranged spirally along an axis to form these structures called strobili, or cones. This is a very organized arrangement. There are two types of strobili. Male strobili, also called microsporangiate strobili, contain microsporophylls bearing microsporangia. Female strobili, or macrosporangiate strobili, have megasporophylls bearing ovules or megasporangia. In Pinus, both male and female cones are born on the same tree. But in Cycas, male cones and megasporophylls are born on different trees, so they're dioecious. The microspores develop into the male gametophyte, which is highly reduced and confined to just a limited number of cells. This reduced male gametophyte is what we call a pollen grain. The development happens within the microsporangia. On the female side, the megaspore mother cell divides meiotically to form 4 megaspores, but only one of these survives and develops into a multicellular female gametophyte that bears 2 or more archegonia. Here's a crucial difference from Bryophytes and Pteridophytes. In Gymnosperms, both the male and female gametophytes do not have an independent free-living existence. They remain within the sporangia, which are retained on the sporophytes. The pollen grain is released and carried by air currents to the opening of ovules. Then a pollen tube grows, carrying male gametes toward the archegonia in the ovules. The male gametes are discharged near the mouth of the archegonia, fertilization occurs, and the zygote develops into an embryo while the ovules develop into seeds. And remember, these seeds remain exposed, uncovered. That's the key feature of Gymnosperms. Finally, we come to Angiosperms, or flowering plants. Unlike Gymnosperms where ovules are naked, in Angiosperms the pollen grains and ovules develop in specialized structures called flowers. And after fertilization, the seeds are enclosed in fruits. That's the defining characteristic. The word angiosperm literally means enclosed seed. Angiosperms are an exceptionally large and diverse group of plants. They occur in a wide range of habitats, from deserts to aquatic environments, from tropical rainforests to alpine regions. Their size range is incredible, from the smallest flowering plant Wolfia, which is tiny and floats on water, to tall trees of eucalyptus that can grow over 100 meters tall. Angiosperms are incredibly important to us. They provide food, fodder, fuel, medicines, and several other commercially important products. Most of what we eat comes from angiosperms. They're divided into 2 main classes, the dicotyledons and the monocotyledons. Dicots have 2 cotyledons in their seeds, while monocots have 1. This is just one of many differences between these 2 groups, which you'll study in more detail later. And that brings us to the end of our journey through the plant kingdom, from simple algae all the way to the complex flowering plants that dominate our world today.
Meet Iyal, your AI study companion. Transform PDFs into smart study guides, practice tests, and flashcards for UPSC, NEET, banking, and any competitive exam.
Master your exam preparation with Iyal.